Hey everyone, Chapter 8 discusses energy metabolism. We're going to look at how our bodies use different pathways to break down all of those macros, your carbs, protein, and lipids in order to extract some adenosine triphosphate. We're also going to look at how our body changes the different types of pathways depending upon which macro it is. We'll look at how our bodies also change in that energy metabolism when it is before, during, and after you eat. And then also we'll look at a few kind of genetic conditions that can alter the way your body metabolizes energy. All right, this slide just gives you an idea of our overall coursework and how it is framed to really start at the very basics of how and why we choose foods. Then we learned about digestion already, and now it's time for metabolism and how all of these kind of streamline our risks of disease or our ability to maintain our health, at least to some extent. All right, just like our lights, in order to keep them on, they need a source of energy. Our bodies also need a source of energy, because you would agree that I couldn't just take a lump of coal and place it in the middle of my house and expect my lights to turn on. Nor could I take a glass of milk and hold it in my hand and have enough energy in order to be able to jump rope. Same thing applies. We have to have something that can actually convert that stored energy or that chemical energy into the energy that we need in order to produce a different type of function. So like coal requires a power plant that can extract that chemical energy. Our bodies within our cells have the mitochondrium, which can really extract a lot of energy. And there's some other areas in the cell that we'll talk about that also generate some energy as well. So our cells have the ability to convert our macros, our glucose, into energy, and this process is known as cellular respiration, and where we use the oxygen that we are breathing in from our lungs and able to convert that chemical energy that's stored within things like glucose into ATP. And here's an example of that mitochondrium, and there's going to be many of them within the cell. And actually, the more you exercise, the more you have that demand for energy, the more mitochondria that you develop within those cells. And they are just kind of our power plants within our cells that are going to be able to convert that energy and release it for our body's needs. Now, metabolism is defined as the sum of all chemical reactions that occur within our body cells that provide energy for the vital processes and the synthesis of new products like organic materials. And so here are a few of those pathways that are involved in energy production. We'll look at the, kind of going first, we'll look at glycolysis first. Now, not every, I would say, definition will actually separate this glycolysis and the acetyl-CoA, because it's such a very quick step. But within this text, it does separate it out, so we'll keep it in that form just for simplicity. Okay, here is another definition. It is energy, which is defined as the capacity to do work. We know that we have energy that's always stored between the bonds, especially those carbon bonds, and in between those phosphate bonds that we'll look at within the ATP. And when those bonds are broken, it helps to be able to generate or release that energy. When we use oxygen within a metabolic pathway, it is known as an aerobic metabolic pathway. When we don't, the opposite of it would be anaerobic pathway. And I want you to know which ones that we define. Are they classified as an aerobic pathway or an anaerobic pathway? Now, a couple unique things to know about metabolism is, one, it never stops. We have a constant demand for it, so we must constantly be able to make it, because our cells don't store energy as ATP. We do have a little bit of ATP within our cells, kind of like our amino acid pools. It's not a storage site, but just temporarily in there. But it is used up very quickly. You see, in 3 to 5 seconds, about the time that would take you to get up from your chair and maybe take a couple of steps, all of that ATP that's immediately available would be gone. So you're going to have to dive into some other processes for generating. You're going to have to go into your power plants there. Another thing is that our processes adapt, meaning that depending upon our own needs and the environment that you're in, like your activity level, your state of growth, if you had some trauma or an illness, if you're just finished eating, or if you are starving, those are two different examples of how your intake can also affect the different types of metabolic processes that we actually use. Okay, you need to know that we have two kind of categories of classifying pathways. Either they are anabolic pathways or catabolic. And the anabolic ones are things that build up. They are after you eat. We have broken down all of the glucose from our macros, and now that we have met our immediate demands, we store the leftovers as glycogen. Or we take those little bitty amino acids from our foods and we make proteins of them. So we're taking something small and we're building it into something big. We eat too much of anything, our lipids, and we can take those and make something that's bigger, which are triglycerides. Our catabolic reactions occur to produce energy. So let's say that we have some glucose in our body, then we can break it apart into something very small in order to create ATP. Same thing, if we need glucose, we have that stored glycogen, so something larger into something smaller, proteins into amino acids, and triglycerides into glycerol and fatty acids. We're going to look at some of these reactions, and I want you to keep in mind and be able to tell yourself, is this an anabolic reaction or a catabolic reaction? Also understand that these are constantly going on at the same time. You are constantly building things, and you're also constantly breaking things down. Anaerobic metabolic pathways like glycolysis, they primarily occur in the cytosol of the cell, meaning that they happen within this fluid part of the cell, outside of any of those organelles. And they don't require oxygen to produce energy, which means that you can really quickly get energy from it without having to kind of pick up your respiration. So it's an immediate source of energy, and this is where those pathways are going to occur within the fluid part or the cytosol. The mitochondrium is going to actually require your oxygen. So it's going to take a little while for it to kick in, but once it does, once we have the energy demands met, we're going to be able to produce far more energy in that fashion. Now we have defined what an enzyme is. They are protein molecules that help to facilitate a chemical reaction. They help things to occur at a faster rate, so without them it would take a lot longer for things to break down. Remember that they're proteins and we can use them again. They're not just destroyed once they perform a job, but they are substrate specific, meaning that they can only take apart things that they were built to take apart and not just act on any type of substrate. So I want you to know the difference between a cofactor and a coenzyme. Our cofactors are made out of minerals. So this, for example, says magnesium, manganese, iron, copper, zinc, calcium, cobalt. Those are all different types of minerals. Whereas our coenzymes, they are made up of vitamins. This NAD plus and NADP, that N is representing niacin, a B vitamin, and the F in this FAD coenzyme is made from riboflavin, also a B vitamin, and these act as electron carriers. So when we look at these different metabolic processes, then you will see within here these little electron carriers acting as coenzymes, and keep in mind that these are made out of vitamins. And so while vitamins don't provide us with energy, as in calories, they do help to facilitate actually extracting that energy. So that's why if you are deficient in these vitamins, then you will not have that energy that you need in order to perform your just basic functions. All right, so we talked about the enzymes, and now it's time to talk about hormones. You've seen these before. We've discussed them in several different chapters. Now I want you to look at how they play a role in energy metabolism. Here's our hormones: our insulin, glucagon, epinephrine, and cortisol. I want you to know which ones are produced by the pancreas and which ones are produced by the adrenal glands right here. So the adrenal glands sit on the kidneys, or they don't necessarily sit on them, but they look like they're right above the kidneys, like little triangle hats. And these are going to be either classified as anabolic or catabolic. You notice that insulin is the only anabolic because it helps to stimulate protein synthesis. You can make protein, and also glycogen synthesis. It does not do anything when it comes to fat. It just helps really facilitate. Now, our glucagon, it is catabolic because remember, it is the hormone that stimulates breaking down things. It breaks down glycogen into glucose. It also breaks down protein into protein into glucose as well. And then it can stimulate lipolysis, or the breaking down of fat. So when we need energy for fat, for example. And then epinephrine secreted from the adrenal glands is catabolic too. It doesn't do anything for protein, but it does stimulate glycogenolysis, which means that it's going to split apart just like this one. It's going to break down glycogen into glucose, and then also just like glucagon, it stimulates lipolysis, or breaking down the fat. Cortisol, also secreted from the adrenal glands, is catabolic, and it stimulates protein breakdown or degradation, and then also stimulates gluconeogenesis. Remember, this is making glucose from non-protein sources. So when I need to make glucose, but I don't have enough carbs in my diet, I can make it from amino acids. All right, now that we have explained metabolism, let's look at exactly what is ATP. ATP is adenosine triphosphate. You can see here in this picture we have the 3 phosphate groups here. We have a lot of energy stored within these bonds. We also have a ribose, or a sugar, and adenine, a protein or amino acid. And so this is the chemical structure of ATP, as long as it has those 3 phosphate groups there. But we know that we can break this apart right here, and we can change ATP into ADP, meaning adenosine diphosphate. So we'll pretend that this phosphate is gone, and that would look like adenosine diphosphate. And that is going to provide us with a single phosphate, a free phosphate, and some energy. One way to kind of look at what's happening when our body requires energy. All right, so we have our glucose, our fatty acids, and our amino acids, all which can generate ATP. Now, once that phosphate group is dislodged or broken from that, or the last phosphate is broken off from the original structure, we have ADP plus, again, that free phosphate group, and energy is released. Now, if we have another phosphate group there within that cell, we can reattach it, and now we have ATP all over again. This can happen to a certain extent, but eventually we use up all the phosphate that is within that cell, and again we'll have to find another route. So if you think about it, we have had free ATP, a limited source that gave us about 3 to 5 seconds. Now we can regenerate some for a little bit longer. We can provide another 8 or 10 seconds worth of energy if we dive into that phosphocreatine. So this is adding in a phosphate group from this PCR, and that is also located within the cell. Our body can use that, attach it again, and then we can go around this route and release more energy. All of these chemical reactions, all of our body functions, everything that we need from our muscles through our kidneys, everything requires this process. So here's a good slide to save and study with. I want you to know that with anaerobic metabolism, no oxygen is required. It occurs in a cytosol, and it can only really provide our bodies with about a minute to a minute and a half minutes when you're going full strength. It is involved, therefore, in high intensity, short duration activities. So think about kind of those HIIT things where you go really hard, real quick for a minute or two, and then you stop. Some of those would be a good example. Some of the heavy weightlifting where you are trying to bulk up more, so you'll do heavy weights and less reps. And so that then you stop and you rest, and that is in order to restore what you need in order to do this process again. So it can happen very quickly, but it doesn't last. We're not going to be able to do long distance at full capacity. Aerobic metabolism, on the other hand, requires oxygen. It occurs in the mitochondria, and it's able to produce ATP pretty much indefinitely as long as we have the fat stores that are in there, and we have a large amount of fat stores on most human bodies. Larger quantities of ATP can be produced compared to our anaerobic pathways. So usually this is taking over when we are at a low intensity and more long duration activity. So even while you're sitting there right now and you're listening, if you're not moving around too much or at a high activity level, your body's using this in order to meet your body's needs. If you were to jump up and try to run out of wherever you're at, your body would have to really rely on anaerobic metabolism. And now we're going to take a closer look at some of these metabolic pathways. Here's a look at some of the major metabolic pathways that we'll be covering in this class. The first stage being glycolysis, the second, acetyl-CoA, third, the TCA cycle, and then last but not least, and where most of our energy will be made, is the electron transport chain. Majority of what we have, the first three pathways here, glycolysis, acetyl-CoA, and TCA, we're going to have some energy that's going to be released here, but we're going to have a lot more NADHs and FADH2s that's going to be made. Now remember that these are the coenzymes, or the electron carriers, that are going to travel down to the electron transport chain here, where most of the ATP is going to be made. Now, all macros can be used to make some amount of ATP, but not all of them can enter into every single pathway. We have here in the very first stage, which is anaerobic, occurring in the cytosol, we can only have the glucogenic amino acids coming from the proteins. We have glucose, and we also have glycerol, which is found in the triglycerides, that can actually enter into glycolysis. This glycolysis also has a reverse pathway that we'll talk about as well. Then in our second stage here, we have ketogenic amino acids. Ketogenic amino acids cannot be used to make glucose, but they can enter into this pathway, which will allow them to be still used as something that will generate ATP and some high energy electrons. Our third stage here, we have acetyl-CoA that enters into the TCA cycle, and it's going to release something known as GTP. GTP is very similar to ATP, so think of it as another form of energy. We also will have some hydrogen ions and their electrons, and then carbon dioxide that's generated from this cycle. And then we'll also have some amino acids that are going to be converted into some intermediate TCA compounds that we'll take a closer look at when we get to this part of the slides. And then in our last stage, the coenzymes NADH and hydrogens, and along with the FADHs, remember that is the electron or the coenzyme made from riboflavin, a B vitamin. It's going to release the hydrogen ions and their electrons in the electron transport chain. And that's where majority again of our energy is going to be created or released. All right, so we're going to take a little bit closer look to our very first stage, which is glycolysis. Glycolysis is a 10-step pathway where we start off with glucose, and we're in the cytosol. This is anaerobic. We don't have to have oxygen to do this. One molecule of glucose, and it's going to go through 10 different steps until it becomes pyruvate, or 2 units of pyruvate, and we'll see that in just a moment. But know that this is catabolic because we start off with this big 6-carbon molecule, if you will, and then eventually we're going to turn out to be 2 3-carbon smaller units here. And this pathway, known as glycolysis, is the first step of breaking down. It's catabolic, but we also have the ability at this point, when we come out with 2 pyruvates, to actually go backwards. Let's just say that our body needs glucose. Well, any of the metabolites that came in that we saw from the previous slide, any of those that are able to enter this pathway can be reversed as long as it hasn't gone on to the next stage. Because once it goes from pyruvate into acetyl-CoA, we can't turn around and go back. But this is an example of how when our body needs glucose, remember that's fuel in everything within our body, Instead of making ATP or energy, what it will do is it will stop it and it will go backwards to create glucose, and that is glucogenesis if it's just coming from carbohydrate metabolites, or gluconeogenesis if it's coming from some of those glucogenic amino acids. So students will ask, do we need to know every single thing that's listed in your tables or diagrams when it comes to the metabolic pathways like glycolysis? And I usually will tell them, what I want you to know is what does it start with? Like for glycolysis, it begins with one 6-carbon glucose molecule like you see right here. And then what does it end with? By the time it finishes just the glycolysis first stage pathway, what is the end products? And so at the very end, what you will see is that one 6-carbon glucose turns into two 3-carbon molecules of pyruvate. We'll also have two net molecules of ATP. So I mean, we're only going to produce 2 molecules of energy here. We'll also have 2 hydrogen ions plus some electrons. Remember, these are the ones that are going to eventually get to the electron transport chain so that you can have additional energy. And we're able to do this because that coenzyme NAD, made from a B vitamin, picks up the hydrogen ions and the electrons to form NADH. This is a oxidation and reduction reactions, and these will help to carry that energy eventually to the electron transport chain. Now, within this equation, we actually make more than this ATP, but this glycolysis step actually it requires energy in order to produce energy. So even though we come up with 4 ATP, it really takes 2 in order to facilitate the process. So kind of like it takes money to make money, just as an example. Kind of same thing applies here to glycolysis. So at the very end, when we kind of check and balance things, we only end up with 2 molecules of ATP. So know these 3, highlight those. So this is a better illustration of what I was trying to talk about. Remember, this is a 10-step process. In this little diagram, you're only seeing the first 3. So we started out, even though it's really a ring, this is depicting it as a kind of linear form here, but it's a cyclic ring of 6 carbons here. And it goes from that glucose down to glucose 6-phosphate. In this process, it actually requires ATP in order to make this next step. And so all we're doing here is we're taking ATP, which remember when we burn up that triphosphate, it leaves us with a diphosphate. And now we have 1, 2, 3, 4, 5, 6 carbons here, and we just added a phosphate group. And in the second stage to the third stage, we're going to go on to eventually create this fructose 1,6-bisphosphate. And what we're really doing, if you look at this picture, we're not getting rid of any carbons here, but what we're doing is slowly adding on a phosphate to each side. So by the time you get through, eventually we're going to have 2 separate 3-carbon plus a phosphate on each side, 2 separate ones of these. It'll be right down the middle. So we ended here from the previous slide with this fructose 1,6-bisphosphate. And in this next part, we're really going to slice them apart. It's a very, very quick step here where we're cutting them apart, and we're going to flip this hydroxyacetone phosphate into 2 of these glyceraldehyde 3-phosphates. And then very quickly from there, we have some of this oxidation reductions where we have NAD. It's going to be oxidized to NADH with the requirements of 2 phosphates. Now we went from just having these 2 3-carbons with 1 phosphate attached to each one. Now we're going to have each one attached with a phosphate group on each side. So we're really building up the amount of phosphates we have in here. And remember, this is what's going to eventually lead to a lot of energy released. Okay, so really until now, when we're looking at these next few steps, is that we have been adding on things. We've been adding on these phosphate groups. And now it's time to start breaking them apart and releasing some energy. So here in 7, 8, 9, very quickly, we have some ATP that's generated and some water that's released kind of as a byproduct. This is where we get some of our water needs met, is through metabolism. And now we have released 2 phosphates. We got 2 more to go. So we have these 2 phosphophenylpyruvates. And then the very last stage here is just to clip off these last 2 phosphates, releasing some ATP. So these are steps 7 through 10, and we've created 4 molecules of ATP. Even though we created 4, it took 2 in order to generate this process of glycolysis. So we have a net only of 2 ATP that's left over. And then we also have the pyruvates themselves, and these pyruvates are going to be able to go on to the next stage, stage 2, which is acetyl-CoA. As I said previously, it's really important to just know the beginning and end product. So this is a good slide to have. At the very end of glycolysis, we have 2 net ATP, 2 NADHs, and 2 pyruvate. And pyruvate is what's going to go on to the next stage. Remember, once it leaves and goes from pyruvate to acetyl-CoA, there's no turning back. It cannot go back to be producing some glucose. It can only go on to some other metabolic pathways like the TCA cycle. For this stage 2, and remember that not every textbook or science course will actually distinguish this as its own separate metabolic pathway, but since this one does, we're just going to stay with it. But this here, remember, now we have left the anaerobic pathways, and this requires oxygen, so it's aerobic pathway. And there's, again, no way for this pathway or these metabolites to go back into the ability to create glucose. So we're starting out, here's your start products, 2 pyruvates. From this point, we're going to eventually create 2 acetyl-CoAs. So all we're doing here is we're adding in a coenzyme A, and we're releasing some CO2, so some carbon dioxide. So this will eventually be exhaled as a byproduct. And we're doing so with the help of 2 NADs, which are oxidized to become 2 NADHs. And these will carry some electrons onto the electron transport chain to produce some more ATP. So starting product is 2 pyruvates, ending with 2 acetyl-CoAs. These CoAs here are made up also with the help of some vitamins that we'll look at pretty soon. So what happens when there's not enough oxygen? And you're thinking, why would there not be enough oxygen? Well, when you're sitting still, your respiration, your exchange of oxygen and CO2 is keeping up with your energy demands. But if I was to just jump up and run into a dead sprint right now, my respiration would take a long time for my body to breathe in that oxygen and then circulate it throughout my body, and to get that oxygen actually transferred to those cells in order to perform aerobic metabolism. So until your body's circulation can actually keep up, then your body has to really rely more on the anaerobic pathways like glycolysis for energy demands. But if you continue to push forward, right, and we have already gone from our 2 pyruvates to acetyl-CoA, then what's going to happen when, again, we can't actually create those 2 acetyl-CoAs because we don't have enough oxygen in there to do so? Well, we're going to have a second type of backup method, and one that you have all experienced before here, looking on our next slide. All right, so a different view here, but same problem. We have pyruvate that's building up to, and remember there's 2, just with one single molecule of glucose, there's 2 pyruvate generated. So you can imagine if we have long chains of glucose that we're consuming, we have a ton of glucose, and then this is going to be 2 units for every one. So it can really begin to build up. And let's say we are really participating in a sprint or something at our full capacity, and our oxygen is not delivered to those cells, there's no way to continue on with this forward. And so even though our muscle cells can't do this, our liver can. And so this is called the Cori cycle. So what our body does is it transfers those pyruvates to the circulatory system until it gets to the liver, and now your liver is able to actually regenerate or use those pyruvates to go back to glucose. And we know if we have glucose, we can go right back down here and produce a little bit, at least 2 ATPs, and then some of those electron carriers in order to generate some more fuel for that very high-paced activity. So this is lactate. With the help of lactate, we go from pyruvate to lactate, eventually to our liver, into glucose, and right back down through the glycolysis cycle again. So this slide just reiterates what we were talking about. Cori cycle is our body's temporary adaptation to supply our energy during low oxygen anaerobic metabolism energy needs, so that pyruvate is reduced to lactate. That lactate diffuses out of the cell, enters the liver, and then the liver converts lactate to glucose, and then it starts all through glycolysis again. So it kind of gives you an idea of that reduction reaction here from pyruvate, changes it to lactate here. We got some electron carriers here. A little bit of energy is going to be released when we go back down through glycolysis again. So again, still here talking about the Cori cycle, but I want you to think of the Cori cycle as your backup energy reserve pathway. You know that if you're going full out at any type of, let's say, race or some competition, there's so far that you can go before you absolutely have to start slowing your pace down. And that is because this anaerobic pathway does produce extra energy. Even though we have run out of the glycolysis pathway, we have that backup here of the Cori cycle. It can generate some of that ATP, but it's not a lot, because we get about 30 ATP from glucose if we have oxygen aerobic, versus just small amounts that's going kind of continually through this Cori cycle. So even though it does allow us to continue with that high intensity activity, it's limited to how much it can keep up with, and that's why eventually we have to stop. We cannot go full out continuously like we can when we drop down our speed. We allow our oxygen to keep up, our respiration to keep up, and then we can go at a lot longer distance at a slower rate. And guys, on this,
during high-intensity exercise, a great deal of energy in the form of ATP is needed to supply hard-working muscles. However, insufficient oxygen is available to proceed to the TCA cycle and then the electron transport chain, as would happen during aerobic cellular respiration. Instead, under such anaerobic conditions, at the end of glycolysis, pyruvate is converted to lactate. For each molecule of glucose that enters glycolysis, a net of only 2 ATP molecules is generated to use for cellular work, like muscle contraction. Lactate then diffuses out of the muscle cells and enters the bloodstream to travel to the liver. In the liver, lactate is converted back into pyruvate. Through the process known as gluconeogenesis, 2 pyruvate molecules are converted to 1 glucose molecule. The glucose is released into the bloodstream, where it can be taken up by the muscle cells and enter glycolysis. This entire pathway is known as the Cori cycle.
All right, so we have left the stage 2 acetyl-CoA. We're entering into the TCA cycle. So these 2 acetyl-CoAs that we start out with, we're going to take 2 cranks of this wheel, TCA cycle, also known as Krebs, or the tricarboxylic acid cycle. And we're going to produce some CO2, some H2O, and then a lot of electron carriers that are going to eventually enter into the electron transport chain. All right, so now we are starting off with our 2 acetyl-CoAs here. We're going to have 2 cranks of this wheel, 1 for each acetyl-CoA. The acetyl-CoA is going to join first with the oxaloacetate metabolite. Then we're going to have some water that comes into play here. And when it does, we're going to have isocitrate. Eventually, it's going to lose a carbon. It's going to form what is known as alpha-ketoglutarate. And during this process, we're going to have 1 NADH and a hydrogen that is produced, represented by these stars here, red being standing for NADH or FADH2, yellow standing for the amount of carbons, and the carbons are going to leave as CO2, or carbon dioxide. And the next step, this alpha-ketoglutarate is going to lose another carbon, and it is going to form succinyl-CoA, NADH, and hydrogen. And then we have the next stage. We have the CoA is going to be removed. We can see that here, right here in the green. We're going to lose this here. It's going to be lost as carbon dioxide. And then we're going to have a molecule of water that's added to it, and it's going to form succinate. Succinate is going to be able to produce, during this process here we have the creation of GTP. Remember I said GTP is very similar up here, representing it, very similar to ATP, so it's a high energy molecule, much like the adenosine triphosphate. Succinate to fumarate, and we're going to also have some FADH2 is going to be produced. Remember that's going to have more of effect on the electron transport chain. And then fumarate is going to be converted to maltate with the addition of some more water here. And then maltate regenerates to oxaloacetate, and we're right back to where we started from. So this kind of gives us an overview, and again, I don't want you to get lost, and no way near for this class do you need to memorize every single name. What you need to understand is what is going into it, and then what are we producing here. We're going to be producing some CO2, we're going to produce some water, we're going to produce also some many different electrolytes here, and some GTP. So a very good slide to again note is that the end products of the TCA cycle here are going to be 2 GTP, then we're going to have 6 NADHs, 2 FADH2s, and then 4 carbon dioxide. This is our major byproducts here for carbon dioxide. So when we're thinking about this, we started out with 6 carbons in our glucose. And by the very end, because we've already gotten rid of 2, we're really just breathing out the remnants of those 6 carbons from the glucose molecule every single time. We just use them to generate things like this. Our coenzymes here are electrons and GTP and ATP. But really the carbons are just being really released by our carbon dioxide. Just as what was mentioned on the previous slide, we started out with the 6 carbons, but then that was found in the glucose molecule. And at the very end, we are releasing those 6 carbons through carbon dioxide. So that's basically our waste products. Our energy that is really kind of added up throughout the whole time is our NADHs and FADH2s. These are carrying our energy all the way through until we get to our next and final stage. And so although there was not any audio within the previous slides, it was just a recap in order to just kind of remind you of some of those very important points. I want to make sure that you know and are able to understand and recall. So back here is the metabolic pathways, the end products. Here we have, by the end of glycolysis, 2 net ATPs, 2 NADHs, 2 pyruvates. These are going to go in and be stored. Remember, we're filling up our piggy bank here of these high energy electrons. At the end of acetyl-CoA, we have 2 more NADHs to add. We're going to have this byproduct of 2 CO2s we're going to get rid of. And then 2 acetyl-CoAs going on into the next stage. And we're going to have the products of TCA cycle, or 2 GTPs, much like ATP. We're going to have 6 NADHs, quite a bit. And then 2 in FADH2s, and then 4 CO2s will be lost in waste. So we really have been storing up this whole time a lot of high energy electrons that's going to be released here within the electron transport chain. Okay, so the mitochondrial membrane, it houses a whole bunch of protein complexes, and these are going to be responsible for creating or generating 90% of the ATP that our body uses throughout the day. And it does so by carrying out a series of oxidation and reduction reactions. And this is when those electrons are transferred from one protein complex to another. And when it does so, it's going to result in forming more ATP in the release of energy plus water. Okay, I don't particularly like the way this image illustrates oxidation reduction actions or reactions, but we're going to go with it. So look at this electron donator here. When it donates or gets rid of its electron, it's known as being oxidized. And then when something picks up something, it is considered reduced, which is kind of backwards thinking. When something gains something, it's being called reduced. And there's a long explanation to this that we won't get into, but interesting one. But just know that when something loses an electron, it's called oxidized. When it gains an electron, it's called reduced. Now, these oxidation reduction reactions happen with every single cycle that we've covered, or metabolic pathway we've covered. So here's an example of that oxidation reaction. We have glucose that is oxidized, meaning that it loses its electron and becomes pyruvate, and we have NAD that gains an electron, or it's reduced to NADH. This slide depicts a kind of a closer look at those protein complexes that are found within the mitochondrial membrane. And they have different colors on here, and these colors often are produced actually by either the minerals or the vitamins that they contain. These flavoproteins here are those mitochondria that contain the B vitamin riboflavin, and the cytochromes are those proteins that contain the minerals iron and copper. And when you know how when iron rusts, it gives this kind of rusty color here. That's what's giving that particular color, for example. So again, remember that our vitamins and minerals, they do not directly provide us with energy, but they do provide us with an ability to extract the energy from our macros. All right, so in this slide, we're zooming in a little bit closer to look at that electron transport chain. So as these NADHs and FADH2s, as they travel through the membrane here of the mitochondria, we have that pressure that is being generated, and it's causing these hydrogen ions to build up within this inner membrane here. So this inner membrane space between our inner mitochondrial membrane down here and then our outer mitochondrial membrane. So we have a lot of hydrogens building up, causing a lot of pressure, kind of like a dam would cause a lot of pressure when that is holding back the water. And when this happens, eventually these hydrogens will go through what is known as ATP synthase. I want you to highlight this, circle this, and know about this protein here. This protein would be like a turbine. We discussed this earlier, how our energy, when we burn it or we put it into water, and then it creates pressure, and it allows us to generate or extract that chemical energy from coal, for example. Same thing. So we have a ton of pressure in here within that intermembrane space, and then eventually as it goes through here, it's actually creating a lot of ATP. That whole pressurized kind of push through is creating a lot of ADP and also phosphorus, and then a lot of ATP here. We also have a byproduct here of water, so again that's contributing to our water intake due to metabolism, and some more hydrogens and oxygen. So this whole time we've really been focusing on carbohydrates and how they produce energy or ATP. We haven't really spent any time looking at proteins or our triglycerides or lipids providing us with energy when needed. So now we're going to take a little bit of time just to look at that. As you know, constantly as we go through this course, you are building upon your previous knowledge. And so we learned about amino acids. We learned how when we need to use the carbons or hydrogens from them, we have to get rid of the nitrogen. We deaminate it. And so once that nitrogen is out of the way and it's used to create ammonia and then urea and excreted through urine, we have the rest of those leftovers here that can enter directly into the glycolysis pathway, and therefore, now that it's here, it can go back up and create glucose, or it can continue down through the acetyl-CoA and then into the TCA cycle to generate more energy. That's only the glucogenic amino acids. Not every amino acid, all those 20, not every one of those can actually do that. Those that cannot do that are known as the ketogenic amino acids, and they cannot enter this pathway, but they can enter in and create acetyl-CoA, and we're going to look at that in a little bit. We also have some amino acids that can enter directly into the TCA cycle. So they enter in and can create some oxaloacetate, as long as we have a little bit of glucose that our body can use. That's a whole nother lecture though. Alright, you do not think that you need to memorize this. I include this for some of my pre-med students because this is a very common type of board question that is a great reference to just save and keep. This shows you the differences between the glucogenic, the ketogenic, and then those amino acids that are both, and indicated by each color here. So you can see when we've been talking about glucogenic, these are the ones that are indicated in red. These are the ones that can enter into the glycolysis pathway but go reverse and really they're facilitating the process of gluconeogenesis to create more glucose. Okay guys, this is a very busy slide, but I want you to pay attention closely here. With this process, we are looking at how our body uses the fatty acids from triglycerides in order to generate more energy. So what you're seeing is that this is a triglyceride. This is our glycerol backbone here, and then we have 3 fatty acids. Remember, they can be unsaturated, they can be saturated, long chains, short chains, medium chains here, but it's just shortening the structure for just of ease of process. So our glycerol backbone here, this is the only part that our body can use from a fatty acid in order to generate a little bit here of glycerol into glucose and energy. So this can enter, this is the glycolysis pathway. Remember, we started way up here, it's 10 steps, and eventually we reach pyruvate. So only these 3 carbons here, following this blue arrow here, can actually generate some ATP, and it can be used to create pyruvate, and we know if we have pyruvate, it can go on to create acetyl-CoA, or it can go into reverse to create a small amount of glucose, but that's not a lot of glucose. Therefore, our high-fat, low-carb diets don't sustain us very long. Now, everything else, these fatty acids here that are linked, when they are clipped off, they can be used in order to create eventually more acetyl-CoA, and therefore it's creating a lot more energy. That's why if as long as you are staying hydrated and you got enough glucose to supply red blood cell formation and to also help to fuel your brain, you could sustain on a lot of your fat stores because we have almost an indefinite supply of these fatty acids. What you're seeing here, though, these fatty acids, when they're clipped off, your body begins to clip them off within equal numbers here of 2s. So it clips off from, let's see, this starts out with 1, 2, 3, 4, 5, 6. In reality, it'd be much longer than that. But it's going to clip off these first 2 with the help of CoA, or coenzyme A. So we're going to have 2 carbons, and CoA attaches it. And then this can go in and create more acetyl-CoA. And then the next 2 will go in. 2 carbons attaches to another CoA, and it goes in to make another acetyl-CoA, or if we need a short chain fatty acid for something else, then we can use it in that form. But they're basically being clipped off by twos, and that's why when you look at our fatty acids, I told you that they are always formed within even numbers. So there'll be 18 carbons, there'll be 20 carbons, etc. And so we're able to equally clip these off, attach a coenzyme A to it, and create acetyl-CoA so that it can go on throughout the TCA cycle and then eventually the electron transport chain. This glycerol here, this fatty acids, this is known as beta oxidation. Everything that we just talked about here following this pathway is beta oxidation. We are oxidizing them and we are eventually creating acetyl-CoA. Know what it starts with? Our triglyceride here, or fatty acids, and it ends with acetyl-CoA, beta oxidation. All right, so this is an overall review, or a very brief and very scant overall review of energy metabolism. So we look at these proteins. How can we make energy from protein? Remember that we have our glucogenic, our ketogenic, and some that are both glucogenic and ketogenic. And these glucogenic down here at the bottom, they can enter in a back door here to the TCA cycle and be able to produce some energy, not a lot, and it has to have a little bit of glucose still within the diet. And then our glucogenic amino acids can enter directly in to kind of the bottom half of the glycolysis pathway, and it can go on either to create acetyl-CoA or it can go backwards to create a little bit of glucose. Anything that enters in here is the acetyl-CoA is not, let me catch up here, is not something that we can reverse and create any glucose from. Remember, this is a permanent, don't go backwards step. And then we also have, see here, our beta oxidation. When we can take that, this is beta oxidation here, but we can also take leftover or excessive amounts of acetyl-CoA and store it as fatty acids, and that is known as lipogenesis, creating lipids or kind of storing your fat. So this shows you when you eat too much protein compared to what you need, your body will take it, convert it to acetyl-CoA, then through lipogenesis create those fatty acids, attach it to a glycerol backbone, store it in your fat cells or those adipocytes. All right, so I think that's about it on this slide. Nope. Oh, just to name this, from pyruvate up to glucose, what is it? Gluconeogenesis. So again, when we're taking these non-carbohydrate molecules and we're creating glucose this way, it's gluconeogenesis. What happens with the other types of monosaccharides, fructose and galactose? Well, they do enter into different parts as metabolites into the glycolysis pathway in order to produce more of that pyruvate, and again can either go upwards to create glucose or back down to create acetyl-CoA. All right, so our metabolism is different depending upon if we have ate recently or if it has been over 4 hours since the time that we have actually put in a macro in order to supply energy. When it is during the fed state, and you have recently ate something in other words, then it is known as the absorptive state. So during this time, you're really breaking down everything through digestion and absorption, and you're going to have a lot more anabolism, that's building up of things, because we're going to store some things as glycogen, right? We're going to build maybe some proteins. So we're going to have a lot more of building up than we are of breaking down when it comes to after digestion. Anything that is left over after our primary fuel needs have been met is going to be stored again as in our adipocytes as extra storage or fuel for later on when it's been greater than 4 hours since we have ate. All right, so when it's been greater than 4 hours since you ate, your body's metabolism is going to be more catabolic, breaking down things, than it is anabolic. So all of these stores like glycogen, from your liver stores and from your muscle stores is going to be broken down to fuel the immediate needs for those cells. You can also rely on some of that adipocytes to break down into fatty acids and release and to meet some of those tissue needs for energy. And then if it's been a lot longer than 4 hours, long-term fasting, your body's going to have to go into some further reserves because we've already depleted all of our glycogen stores, our carb source stores for energy. Now your body's left to really start breaking down structural proteins, and we don't want that to happen. So we break down those amino acids. Some of those can be used for glucose. Remember those glucogenic amino acids. We have some of our fatty acids that can be used to meet our energy needs. And then also our liver can take some of these fatty acids and create some ketone bodies to at least nourish our brain cells. This is not an indefinite process here, remember, because eventually we're going to need to create more red blood cells, and this will not be able to fuel that. All right, I've covered a little bit about ketosis. Ketogenesis is defined as the formation or creation of ketone bodies. This occurs in our liver mitochondria and only due to prolonged low carb or insufficient carbs within our diet. It could occur in starvation and can occur in diabetes or insulin-dependent diabetes. Ketosis generally peaks on day 4, and this is when people who are experiencing this will complain of the keto flu. Remember that ketosis, nutritional ketosis, is not the same as ketoacidosis. This is what someone, especially who is type 1, has type 1 diabetes, would experience because their body does not produce any insulin. 30% of the fuel from ketones and about 70% is going to be fueled from our blood glucose. Oxaloacetate eventually becomes depleted in our liver, so we cannot use oxaloacetate in order to meet our TCA cycle continuously if we completely deprive ourselves of glucose, because we do need a certain amount in order to be able to turn that cycle. So low levels of that oxaloacetate prevents our acetyl-CoA from entering the TCA cycle. Our acetyl-CoA builds up and then is converted to ketone bodies. Then within those ketone bodies, just to kind of give you an idea, one of those ketones is known as acetone. Acetone is what you would know as fingernail polish remover, so you can smell that. That is a type of ketone, and that is what can, if necessary, fuel our brain cells. But you can see how if you don't have a little bit of glucose and this begins to take over the majority of your energy production, how acetone can cause your body to go into ketoacidosis. Again, this cannot happen if you have a functioning pancreas. So nutritional ketosis will not cause the ketoacidosis. Only someone who does not have insulin production would actually have that ketoacidosis. And in this form, this right here, is what creates the smell of kind of a sweet acidic smell that someone who has very high blood sugars, who has diabetes, would kind of give off. It's a fruity acidic smell. All right, so these last few slides are just going to cover a few of the genetic diseases that are out there that can affect metabolism. The first one being a little bit more well known is phenylketonuria, PKU. It's when our body does not have the ability to break down phenylalanine because we lack the enzyme that is required to break it down. Just like someone who is lactose intolerant does not have enough lactase, the enzyme necessary to break down dairy. Someone that has PKU is missing phenylalanine hydroxylase, which is the enzyme that's necessary to break down this amino acid. And aspartame is made from the amino acid phenylalanine. And so you have to learn to avoid these foods that are made using any type of aspartame. If you do not adhere to this diet, it can lead to a lot of mental disabilities, behavior problems, eventually coma, seizure, and death. The next one is called maple syrup urine disease, MSUD. This is an interesting one. This is when our body does not have the ability to break down our branched chain amino acids. We didn't go over what were the branched chain amino acids, but they are valine, isoleucine, and leucine. These are all essential amino acids. You will definitely need to know this if you are pre-med. These are known as the villains, valine, isoleucine, and leucine, villains. These are a branched chain, and they require again, alpha keto acid dehydrogenase in order to break them down, and someone who does not have those, it causes a buildup of these types of amino acids, and it leads to your urine being the color of maple syrup, but also very sugar-like. And in fact, that was the way that they discovered this, is that people who, or I guess guys who would pee outside, ants were attracted to it because the metabolite is very sweet. So where are these amino acids? Well, of course, any complete protein, so any animal protein is going to be in there. And so they have to go on virtually a low protein diet, and therefore they have to have these specially designed formulas that still provide enough amino acids but are lower in valine, isoleucine, and leucine. They also have to have glucose infusions so that your body does not begin to go into more gluconeogenesis than it needs to. Homocystinuria is another metabolic genetic disorder, and this is when your body has the inability to break down homocysteine into cysteine. Now this here is kind of a limbo. So your body has, of course, amino acids. Methionine is one of those essential amino acids. When your body wants to create cysteine, a non-essential amino acid, it goes through these steps here. Well, if you don't have enough of this particular enzyme here, you don't need to know it by name. If you don't have enough of this enzyme, well, it gets stuck here, and so you have a buildup of this particular metabolite, and again, anything in excess than what it should be becomes toxic. It can result in things like eye disorders, blood clot, atherosclerosis. In adults, it creates a higher risk of cardiovascular disease. So people that do have homocystinuria will have to go on a diet that is low in the amino acid methionine. And the next one is galactosemia. You have any type of the monosaccharide that is used to create lactose, of course. Your body can use that to create glucose and then ATP, and it can also be used as a metabolite eventually to create glucose. But if you have galactosemia, you do not have the enzyme in order to finish this process. And so again, you have a buildup of this galactose 1-phosphate. It becomes toxic, and it ends up causing a whole bunch of things, starting out with nausea and vomiting, failure to thrive or grow. It can lead to mental disabilities and death. So, of course, anybody with galactosemia has to avoid the major source of galactose, and that would be dairy. And now we are at the end with glycogen storage disease, and this is when the body is unable to break down our stored glycogen in order to provide our bodies with glucose when we're not eating. And so our glycogen begins to build up within our liver, which definitely, once we fill it up, can be dangerous. And that means if we have all this stored glycogen, but the inability to break it down, we're not going to have glucose in order to keep our blood glucose in a homeostasis or balanced level. Again, that leads up to a metabolite buildup, and that leads to low blood sugars, it leads to seizures, a high uric acid level, hyperlipidemia. Of course, it can get worse than that and lead to more things affecting the brain, coma, seizures, and death. Anybody with glycogen storage disease is going to have to avoid really high carbohydrate foods. Anything with sucrose, lactose, galactose, you've learned the monos and disaccharides, so you get the idea here. All right, now if you're looking within your textbook, you will see more than just the ones that I have covered here. So you do not need to know the ones that I have not covered within the PowerPoints. Same thing along with the rest of the information within the textbook. This is a little bit more condensed version of Chapter 8 than what is included within your textbook. All right, so this brings us to the very end of Chapter 8. Have a good day.